And people who were working in tumor systems also made the similar observation that PD-1 is a major target on the TILs, these tumor-infiltrating lymphocytes that get into the tumor but cannot kill the tumor, have very high levels of PD-1. Then there was a lot of interest in extending these in vivo experiments from mice into non-human primates and so on. So actually we did a study with Rama Mara, who's also a faculty member at the Emory Vaccine Center, and Rama showed that if you give an infusion of this blocking antibody to monkeys that were infected with SIV, SIV stands for simian immunodeficiency virus. It's a cousin of HIV, and basically it's the HIV virus of monkeys. And what Rama and his group showed is that if you block anti-PD-1, PD-L1 interaction in non-human primates, you get an increase in the function of the SIV-specific T cells and a reduction of the virus in these monkeys. In a collaboration we did with a group in Ohio, with Chris Walker, we showed the same thing in the HCV model of chimpanzees. That's really the only model for HCV that can be done. And then Chris showed the same thing happening in HCV-infected chimpanzees. And as I mentioned, a lot of people working in tumor models showed the same thing. And then what has been quite amazing to see is the very, very, very rapid advances that have happened in terms of clinical trials of PD-1 in cancer patients. This, of course, is not our work. We don't work in cancer models. We actually never even worked in a mouse cancer model. Okay? But the real benefit of PD-1, and the progress has been quite remarkable, has happened in the last 3 or 4 years. And these studies have been led by many major pharmaceutical industries, but 3 in particular, Merck, Bristol Myers Squibb, and Genentech. These 3 big pharmaceutical companies have developed anti-PD-1 antibodies and also anti-PD-L1 antibodies. I told you both, in effect, do the same thing. They block this inhibitory interaction and release the brake on the T cell. And the results have been quite promising. PD-1 blockade has been shown to be effective in both phase 1 and phase 2 clinical trials for multiple different cancers, in particular for melanoma and for lung cancer. In particular, lung cancer really had hardly any if therapy was there in terms of chemotherapy. All of that was really not that effective. But depending on, but the PD-1 has given promising results in 15 to 20% of the treated people. That's not a very large number, but when you go from basically no survival, 0% survival, to 15 to 20%, that's really quite a dramatic change in the treatment of cancer. And within 3 or 4 years of these human clinical trials starting, both Merck and BMS have a licensed drug. So PD-1 was licensed last year for melanoma treatment, actually a year and a half back, for melanoma, and last year also for lung. So this has been quite remarkable to see, and I give this as an example for how science should be done. When we first started down this path many years ago, 25, 30 years ago, really interest was very simple. We asked, how do you maintain T cell memory long term? We were not thinking of cancer, we were not thinking of immunotherapy. That was completely out of our radar. But these fundamental studies on understanding T cell memory led to T cell exhaustion. The T cell exhaustion led to the linking of PD-1 with T cell exhaustion, and then a potential therapy for the rescue of those exhausted cells. And that has very quickly really work done, and the credit for this goes to the oncologists who did these clinical trials at different places, these companies who moved things very forward very quickly. But essentially you now have drugs which are effective in treating melanoma and also in treating lung cancer.